Okay, hello everyone. You should have gotten the notification now that I start recording, so if you don't want to appear, basically don't say anything. Thanks for being here. And today we have Yann Fichou. He studied physics in Rennes in France. Then he did a PhD at the Institut de Biologie Structurale in Grenoble, also in France. And now he's a postdoc at UCSB with Song Han, but actually right now he's back in France. And today he's going to talk about DEAR on singly labeled protein as a new tool to characterize amyloid aggregates. Yann, the stage is yours.
Yeah, all right, thanks. Thanks, Nino, and thanks everyone. It's been a great meeting, I wanted to say again, so I'm glad we're having all those virtual meetings. So today I want to share some results, what we think is an original way of characterizing amyloid aggregates with DEAR spectroscopy. So first of all, what are those amyloid aggregates? They're highly ordered protein aggregates where each protein stack onto each other in a cross-beta sheet arrangement. And because all those proteins stack along the same axis, they form those filaments here that are called amyloid fibers, amyloid fibrils. And you have a computer modeling here of those fibers. And amyloid aggregates are heavily studied because they're involved in many diseases, and in particular in neurodegenerative diseases such as Parkinson's disease, ALS, Alzheimer's disease, and others. Now the protein I'm most interested in is the tau protein, which is an intrinsically disordered protein, meaning that it doesn't have any unique 3D structure, but rather exists in an ensemble of conformation that you can see here. It has several isoforms. Let me actually try, let me know if the pointer is too messy. It exists in several isoforms. And the longest one is 441 amino acids here. But most of the result I'm going to show is from these fragments of 187 amino acids, which contain those 4 repeat domains that mostly contain the amyloid core forming those fibers. So indeed, tau can form amyloid aggregates in a class of disease that you call tauopathies, among which the most well-known is Alzheimer's disease. Now, to really understand the complexity of those aggregates, there is one more thing we need to know. And this is that if you take a slice of a perpendicular transversal slice of those fibers, and you look at the conformation of the protein can take many conformations. And actually, even tau itself can adopt many different conformations. And here you see 3 of them that were solved from brain tissues of different disease. And so this is so important that actually this structure can be a diagnostic of the disease the brain is affected by. So there is a question that we as a group and many other people are really interested in is what type of aggregation pathways lead to the formation of those different configuration, different conformations, and what are the molecular determinants to reach a given structural selection. So with this question in mind, we thought that DEAR actually could contribute in several ways, as you will see. So the most conventional way to use DEAR, so to say, is obviously to have 2 labels in your protein so that you can measure intramolecular distances. And with that, you measure dipole coupling that you can relate with the distance between your 2 spins, and that gives you your intramolecular distances. Now, you see we have a fiber. So the way in practice we do that is by using a mixture of labeled protein, a small amount, and an unlabeled protein before triggering aggregation, so that the labeled protein get diluted along this axis here, and you mostly measure this intramolecular distance. And when you do that, still obviously you have those inter-pair interactions, which often call background because you don't want it, and so then you background correct and you remove it so that you're left only with your dipole coupling from the intramolecular distance. So the point of the point I want to make today, and you'll see, is that this so-called background actually might carry some interesting information on the fibers. And I will get to that in a minute. But before, I just want to show one example of how you can use the intramolecular distance to answer some question for the amyloid aggregates. So how do we study aggregation? Well, there is an obvious first step, which is to be able to make fibers in vitro. The way we do that with tau is by adding cofactor and most of the time heparin. And when you add it to tau, you have spontaneous aggregation, as you can see by thiophallavin T fluorescence, and you can verify by electron microscopy that you have those amyloid fibers. And so the first question we wondered was how close those in vitro fibrils that you make in your test tube are from the structure that were solved recently in the brain. So to answer this, the design of the experiment is such that we place several labels, obviously 2 at the same time. So each of those distance is 1 different mutants, 1 different sample, so that you can probe some key distance. For instance, this one will probe the C shape. Also the beta solenoid could be probed by this. And what we did here is we measure those different distance for the heparin fibrils, so for our model, and you see what we measure in black here using a pretty simple Gaussian model. And in red and blue is the distance that we computed using the MTSL rotamers based on those conformation that are from the brain. So what you can see pretty clearly here is that those heparin filaments are actually very far from having the same structure that what you have in the brain. And in addition to that, actually, you see that those heparin filaments are heterogeneous because of the width of your Gaussian that are usually pretty broad. So this well, was published 2 years ago, so I didn't want to spend too much time, but I thought it was interesting to mention it. And now I'm back to the type of signal that we obtain. And as I said, I hope to show that actually the background might contain information. And for that, I'm going to say a few more words about this background equation. And I think most of you have seen that, but maybe a fewer actually knows where it comes from and how we can interpret that. So let me spend a few minutes on explaining what it is. So first of all, this equation only holds for randomly distributed spins. That's pretty important to understand. It has 2 parameters, alpha and d. Alpha is related to your spin density, which is the concentration of spin in your spin bath, as well as the pump efficiency, which means the probability that your pump pulse is going to flip the spin. Now, if d equals 3, and I will come back to what it means, d equals 3, you have pretty simple analytical equations where I think what you have to remember is that the concentration is proportional to alpha. If now you have alpha, any alpha, you have a slightly more complicated equation here, but I guess what you should remember again is that you're still proportional to your concentration, and that's pretty interesting, pretty important. Now, what about D? So D is the so-called dimensionality, which represents the dimensionality of your spin bath. So D equal 1 will give you a line, 2 a plane, and 3 a volume. And I should mention that accordingly, the concentration that you have inside your alpha depends on your D. So if you D equal 1, then your concentration is in number of spin per length. D equal 2, you get a concentration number of spin per surface, and etc. So now there is a very important point I want to make, which for me was pretty hard to understand, is that D is not related to your the orientation of your spins, which means that if you're this spin here, it's not because you see only neighbor on the left and on the right, or if you see neighbor everywhere on a plane, that you're going to be D equal 1 or D equal 2. And this from the math, it's obvious because anyway, this equation up there apply the powder averaging where every orientation is averaged. So there is no need, but rather where what this D is coming from, it originates from different distance distribution that themselves are different because of those different conformations. So let me show an example. I know this is not clear, but I want to spend some time because I think it's very important to understand. So here you have those different configuration, and what you can do from those configuration is you can compute the distance distribution of your first neighbor. And you see the shape is very different because you have those different 3D, 1D, 2D. Then what you can do is, based on this one-dimensional P of R, you can compute the corresponding dipole coupling by only involving 2 bits, 2 spins. So it's a very simple dipole coupling where you calculate your signal at different distance, and then you do weighted average weighted by this distribution that you have here. So you get a signal that you can fit the same way that you will fit an experimental curve, and then you have an output parameter fit that is D. And you see that here we're finding D equal 1, 2, and 3. So this is very important because it means that it's really this P of R that give you your D, and it's not directly your volumic or linear configuration. And the implication of that is that if you have this D equal 3, for sure you have this P of R, and for sure you have D equal 3. But if you measure a signal and you can fit it like this, D equal 3, then you might reasonably get back to the P of R like this. But it doesn't mean at all, or it's not at all straightforward that you have this configuration here. So this is a one-way travel. And that, I think that's very interesting. So with that in mind, let me now switch gear and show some data of what we did with that. So here is the first set of experiment where it's kind of we measure free radical. It's kind of a test measurements where we have free radicals in solution, and we change the concentration of this radical. It's tempo, 4-hydroxy tempo. And we fit it with the equation, and we extract the dimensionality that fits and the alpha that fits. So first thing to notice is, and it's always good to verify that your theory works, is D equals 3 pretty consistently independent of your concentration. And here you start having deviation at high concentration, and this is because of exclusion volume, where because you're too crowded, then your tempo can't overlap. So you actually break your assumption of a random distribution. Therefore, you break this equation, and you can see the fit here for this high concentration actually doesn't fit very well. Second thing to look for is that alpha is completely proportional to your spin concentration. And so if you look at this equation, what it means is that PB, which is the pump efficiency, is actually independent of your sample. It's independent of the concentration of your radical. And then what we can do is we can actually, by measuring alpha, we have a complete readout. We have a direct measurements of your spin concentration. And for instance, if you have a protein in solution of a known concentration, then you can measure spin label concentration, and you will get a spin labeling efficiency. I know usually you do that with quantitative CWEPR, but for in some condition, this is actually a convenient way to get some information. All right, so now let me switch back to my my fibers. So my object of interest. So here we have the fibers, and what we do to prepare those is we mix, as I said, we mix a small amount of labeled protein and unlabeled protein, and then we trigger aggregation so that hopefully the spins are randomly distributed here, like this. Here in this experiment, we vary the ratio of unlabeled to labeled protein, which effectively change the percentage of spin label that you have along these fibers here. And so we fit it with the same equation, and here are the output parameters. So the first thing that really was striking to us is that D is really stable. It's really independent of your dilution factor, of the quantity of spin label you add in your fibers, and it's always equal to about 1.5. So it's much lower dimensionality than 3, and it's pretty consistent at 1.5. Second thing to notice is that alpha actually is pretty well proportional to your percentage of spin label. So what this means is effectively alpha can measure the spin density of your spins inside the fibers. And this dimensionality most likely originate from the geometry of your fibrils, this number here, and I will come back to that a bit later. So now how do we use that? Well, here's one example to use in particular this relation here, the capacity to probe the spin density in the filaments. So one more thing to know about those fibers is that sometimes they exist like this, where you have only one protofilament, which means that you only have one monomer per slice per layer. But you can also have two protofilaments where you see you have two of those, well, they're called protofilaments, together forming the fibers, the filaments. And if you take a slice here, you actually have two monomers forming these fibers. And making the difference between those two is extremely difficult, meaning knowing if you have one or two protofilaments in the fibers you're looking at, even by EM, is extremely difficult. So now what happens if you spin label? So the same way, you dilute, you have a little bit of labeled protein, some unlabeled protein. And you do that for this. And what's interesting is that if you have a single protofilament here, you end up with a density like this. So you see you have 3 spins up there. But if you do it with 2 protofilaments, then because you have 2 monomers, you actually have double spin density along the fiber axis. So you have 6 spins up there. So without, and I guess you know where I'm going, is that because we can measure spin density, in principle, we are capable of making the difference between those two. So we went ahead and we measure 2 different fibers, one from the Taiwan 87 I mentioned, and another one from a small peptide, 16 amino acids here. We made fibers with the exact same dilution of labeled to unlabeled protein. So we had 6% of spin labeled protein inside this mixture. And yet we saw, so there we see the signal, we fit it, and you see that D is again fairly stable, around 1.5, 1.6. But alpha is pretty different, and alpha of the tau 16 sample is really high compared to tau 187 when the spin label dilution is actually the same. So what this means is that this sample is a higher alpha. So it's actually, you have 2 protofilaments in there. So you have higher spin density. And because this is still a proof of principle, we actually know what this is making. So we know that this is making this type of conformation where you actually have 2 monomers per layer. So it's consistent, and therefore the other tau 187 is lower alpha. So it means it's going into this class. So you have 1 protofilament as it was conjectured before. All right, so another application another things we explored, let's say, was to spin label at different position. So each time it's a singly labeled protein, but just different mutants, where we move the spin label along the protein. And again, we measure the same signal that we fit, and we observe dimensionality here. So for those 3 sides, you see that it's again fairly stable, 1.5, and there's only one outlier, that is 404, and which is the spin label in the C terminus of the protein. And again, because we're trying things with this protein, we actually have a little bit of knowledge of this protein. So we know that those sites are inside the amyloid core, so meaning it's inside the location that make those cross-pair sheets, while the 404 is toward a place where it's still disordered even inside the fiber core, forming the so-called fuzzy codes. So what this is telling us is that D seems to be sensitive, seems to be able to tell us the environment of the spins of the residues that you're looking at and tell us whether you're inside the core or outside. Finally, the last piece of data, which is still preliminary results, but I thought I'd share it, is the capacity to measure time-resolved aggregation. And so for that, we took one sample that we measured before inducing aggregation, just 12 minutes after, and toward the end where the THC signal is almost maximal, and actually one more point at the end. And again, this is preliminary. There is some more work to be done here, but I hope you can appreciate that there is consistent change, and you can already make one conclusion. So here you're jumping from a high dimensionality around 3 because you are 0 minute, meaning you're a monomer in solution, to lower dimensionality. And your alpha is also being from very dilute, and this is because you're 3D, so you're in solution, so spins have space. And here you increase your alpha, which means you increase your spin density already at 12 minutes. And what that means is that you actually have oligomers pretty tight together, actually, that are already formed at 12 minutes, although the THC activity was still pretty low. So I think that's also a pretty interesting application of this. Now, I'm reaching the end of my time, so there's still one thing that I haven't mentioned is this D equal 1.5, and you see that 1.5, 1.6, it's really consistent. And why is that? So if you had a linear fibers where all your spins are aligned, you should actually get down to a D equal 1. So we use simulation to understand, try to understand where was this number coming from, with especially the help of ASIF to do this modeling and simulation. So to do that, we try to model an amyloid fibers in the following way using some type of, you could call it the 1D Monte Carlo simulation, where we have bits. We have a few millions of bits like this. They're spaced by 4.7 angstrom, and each of those bits has a probability to be paramagnetic. And so you fill your line like this with yes, no, paramagnetic. And essentially what you're doing is you're exactly, your probability of having a spin is exactly your spin dilution, your ratio of labeled protein to unlabeled protein that you use to make fibers. So if your P equals 0.1, for instance, it means it's equivalent to 10% of spin labeled protein. So we did that, and from this configuration here, we computed the first neighbor distance distribution that you have in this line here at different probability P here. And you see that you have different P of R. And one more time, what ASIF did is, based on those P of R, is simulated the dipolar evolution signal that we will obtain by just having the different oscillation with different distance and weighted with the distribution that were calculated here. And from there, you get a signal that is a fake measured signal, if you will. And then you can fit it with the same equation here and obtain the D and alpha. So consistently to what you would expect, in a sense, you find D equal 1 and independent of your spin density, so independent of P, while alpha is proportional to this. So what this is telling us is one, well, it's working, and two, considering the first neighbor seems to be enough to match what you expect from the theory. So yet we don't understand why it's 1.5. So we thought let's consider the different deviation from this ideal case that could be experimental. And one of this pretty obvious deviation will be the fact that your label has some intrinsic motion. So it can rotate itself, right? So even though it's anchored to something aligned, it can rotate, but even more, the sidechain can move, and potentially locally, you have some motion, some mispacking, or some flexibility, so that your spin is not on a line, but it's evolving around here a disk. So it's a perpendicular disk here. That's where your spin is allowed to move randomly. And the amplitude of this disk, which is the amplitude of motions you will model, can be changed. So we did all that. We again computed the P of R for perfect alignment and different amplitude of deviation from this alignment. And you see the P of R is quite drastically influenced. And here we extracted from fitting those data the D. And you see that past a certain deviation from this line, you start increasing D. And according to those numbers, if we want to reach the experimental number D, you will, if you want to explain it only by this deviation, then you will have an amplitude of motion of about, let's say, 7 nanometers, which is still big, but you know, at this point we're mostly qualitative. So another deviation that can happen, and that's I'm reaching toward the end of the presentation, is if your label are not completely randomly incorporated inside the fibers, but they rather have interactions, either by attracting themselves. So if you stack a label protein, then you're more likely to stack just above another label protein, or some repulsion, which I think is the most likely because your labels take some space. So if you have a label protein at a position, then the other protein that come on top is more likely to be unlabeled because you have less space for the label. So we model that by playing with this probability of having a spin in a way that if you have a spin at this position here, then the chances of having one in the neighboring space are changed, either lowered if you want to mimic repulsion, or higher if you want to increased if you want to mimic attraction. So we simulated those cases and going through the same workflow, a P of R, simulation, fitting, and extracting D. And here you have the data here where, so the x-axis gives you how far away this interaction is taking place. So are you having an interaction on the first neighbor, on the first and second, on the first and second and third? And this is the amplitude of your interaction, whether this is attraction, so whether the chances of having a neighbor is 100% higher or 50% higher. And you see here that the trend on D is, well, it's the wrong way. If there is a trend, it's going down, and there is a trend. It's going down, but even 3 neighbors actually make very little difference on the apparent D. So no repulsions, that I think is more likely, because repulsion means the spins don't want to be close to each other, and you can understand that is a steric hindrance from the MTSL. So same thing, whether this repulsion goes over 1 neighbor, 2 neighbors, or 3 neighbors, and whether you completely forbid, it's the full circle, you completely forbid to have a neighbor, or you just decrease the probability by 50%. And so here you see that you have a trend that does increase D, but even over 3 neighbors each direction, so it's pretty long range interaction, you still are pretty weak compared to the experimental value. So it doesn't mean that those interactions don't happen, but it means that it can't be solely responsible for the D equal 1.5. So which means that we're really we're thinking that the label motions is a key part in interpreting this D equal 1.5. All right, so I'm quite over time, so let me finish here. I think the take-home messages that I wanted to share today is that really this dear so-called background, right, actually carries information in our case of amyloid fibrils. Then we can measure with that the spin density in the fibrils, and we use that to detect the presence of protofilaments. The dimensionality inform on the geometry, qualitatively on the geometry of your spins, and mapping potentially the sites in the core. And finally, using the first neighbor distance distribution, we could make a simulation that recapitulates D, and that can help to interpret the observable, in particular suggesting that the flexibility of the label is measured here. And with that, I thank you for your attention. I thank all the group, the Songi's group, Songi Han for nice, really good mentoring, and Asif, Iqbal, and Karen Say, which who has been instrumental in this work, as well as Gun and Jeske for email discussion that were really interesting. And I'm happy to take any questions.
Thank you, Jan. So yeah, you can, again, as always, raise your hands in the participants panel. I will also allow you to unmute yourself now. Okay. So any questions? Jan. Tufa?
Hi, yeah.
Hey, Tufa.
My question was, so the 1.5 dimensionality, of course, you probably would expect a question on that. I was thinking that it may have been a mixture. I thought it was very interesting, by the way, but I thought it might be a mixture of a 1D matrix with a 3D matrix, right? But I'm guessing at the start, you were showing that you were diluting spin, you're doing spin dilution, and you didn't find a difference, but maybe like you would need to like dilute even further, like 1 to 100 or something like that. Like, did you consider that concentration effect?
Yeah, absolutely. So no, that's absolutely true. That's also what I thought originally, and I can tell you it's not the case for a few reasons. First of all, if you can easily not even simulate, but you can just add a 3D, the equation for 3D and the equation for 1D, if you have a mixture of perfect both 1 and 3D, and the signal is different, the shape is different, and you can no longer fit it like this. So yeah, you might see it, but let's say maybe deviation is not so big, so that within the noise you can see it. But in that case, still we've changed concentration, and you see D is consistent. When you change dilution factor, the fact that alpha is proportional is also a good indication. And maybe the ultimate proof is that you can actually, with biochemistry, you can actually make sure that you no longer have a significant amount of spin in solution. And maybe I should have started by that actually. But yeah, we have about 95% of our protein included inside those fibers.
All right, cool. Very interesting work.
Thanks a lot.
Okay, more questions? Don't be shy, Hassan.
Hi, it's a very nice talk. Just a quick question. Did you have done any relaxation time measurement? Just to see if there is any correlation on what you have done with the relaxation time.
So like T2 measurements. Yeah, we haven't done. So well, I always record it anyway, and I use it as a way to reassure that what we're doing, we're consistent. So if you look at T2, for instance, you see that the T2 relaxation correlates with the alpha that you measure with the spin density. So well, it correlates with the alpha that we measure and that we interpreted with as being a spin density. And then it makes sense because T2, as you get your spins closer and closer, especially because this is very tight assemblies, then you will expect that you're losing your T2. And if you go to low dilution, so a lot of spins in your fibers, really you almost get no signal anymore. So we use that as a way to say, yes, we are actually measuring spin densities, but not more than that.
Okay, thank you.
Okay, thank you. More questions?
Hi, Yann. I have a question.
Hi.
Yeah. So, is it possible that the dimension 1.5 is a result from protofilament?
Yeah. No. Yeah. That's a really good point, and that's something I haven't shown because it's too. So we're simulating that to see how having those protofilaments are changing the dimensionality. It's possible, yes, but at the same time, so it has. My point is, it's in principle it can have an effect on D. Then for our case, the tau fibrils most likely have only one protofilament. That's what I showed in when I was quantifying the spins, and that was shown by a previous structure. So, which means that in that case, you should get a D equal 1 if the protofilament were responsible.
Okay, thank you.
Okay. More questions? Actually, I have one. So now, back to when you do this kinetics, I mean, at this point, you must have basically different dimensionalities, right? So if you aggregate, you have some in 3D and some in the amyloid. But I guess you fitted 1D for the whole trace.
Yeah, no, that's correct. So that's where, yeah, go ahead.
Okay, I mean, the next question is, shouldn't you basically have two different dimensionalities with the population or something like this?
Yeah. So yeah, no, yeah, I agree with you, and that's why I wanted to share that because I think it's an exciting perspective, but interpreting that is much harder. So what I did there is I quantitatively, simulate, so to say, mathematically calculate at least a mixture of 1D and 3D at every 5% from 0 to 100%, so I can see how the shape differs, and so yes. So the short answer is yes, we have a mixture here, okay. And ideally you'd like to involve 2Ds and 2 alpha, but then you'll be clearly overfitting and you wouldn't get anything. So I think what you can say for sure is that this big increase in alpha is clearly a sign that you're clumping, all your spins are getting together. Then I'll be much more skeptical of interpreting D values for as a relevant of your system. This is much harder because you have a mixture, but even when you mix, for obvious reason that you can imagine, because even if you only have, let's say, 10% of fibers, because they're so close to each other, because they're aggregated together compared to the 3D bath. So this is going to have a huge contribution on your alpha. So you're actually sensitive to the species where your spins are close to one another. But making a quantitative interpretation because of this mixture will be to be taken with measures.
Yeah. It might come back a little bit to the question, right, of Hasane with the relaxation. Depending on how you choose it, you might kind of probe only the one, like one of them, or probe one more than the other. But still, the trend is still clear. I mean, it's not.
Yeah, no. I mean, I think that's what I said. It's preliminary, and I think one could push that a bit further, and we haven't yet, at least. But no, I mean, I don't know if Hassan had a precise experiment in mind, but yeah, those will be. I mean, if you wanted to jump in, Hassan, and say if you had something in mind, but yeah, no, you're right. There will be more to be done.
Sorry. Can I?
Yeah.
So we have seen in some cases that the relaxation time, the TM, can give you some indication about, but just by looking to the slope of how the decay is, that can give you some indication about differing rates in the relaxation time, which means that you do have different domain, like highly concentrated that is very fast, and then you have very slow decay, which means that now you start to move to nodal domain. So that's why I was wondering if you have done any kind of correlation between what you have found by using DIR and the relaxation time.
Yeah. No, you're right. Yeah, you're right. That's a good idea. And also because the relaxation, you can, I mean, in principle, you can have, you can fit, it's not stretch exponential. You could use a regular exponential, and then you can fit more of those exponentials into the signal. Although, I mean, I know it comes from different phenomena, but with some assumption, potentially you could fit 2 species of contributing to relaxation. I don't know if that's well, yeah. But I get the point. I think it's good.
Any other questions? I don't see any.
I can ask one if you want.
Yeah.
So how did you make the aggregating tau fibril? And do you know beforehand which one it is? Alzheimer's pick or whatever, you know?
No. So what we showed, it's none of those. It's none of them. So that's what that was the so-called the kind of introduction I made. So that was the point we made in 2018, and then actually 6 months after the full cryo-EM structure of heparin fibrils came out, and I mean the title was essentially the same. They're different, heterogeneous and different from any of the brain structures. So at this point, we're not making any of those brain structures, but it's a race we're pursuing for sure.
And I guess you can work with it easily. You're not going to die from toxicity of tau, right? Because with PRP-SC, we have the problem that it could attack us and kill us.
Yeah. I mean, that's a really good point. And you know, I've been scared, and sometimes I'm still scared because we're talking of I mean, it is called a prion-like protein. It's being called like this. What saves you, I think, is that it hasn't been shown to pass the brain-blood barrier.
Right.
Which, you know, for myself, I'm convinced. I'm being convinced of that, because otherwise I would stop working in the lab, because we're still assuming in level safety #1. So.
Well, you'll be your brain would.
But yeah. If I start forgetting stuff in 5 years, then I know where it comes from.
I mean, in our lab, we just have a lot of people who have been on our lab who haven't got symptoms or anything like that. So I think that's what we work with PRPC, but and it's also like mouse PRPC, so it's like species barriers and so on.
Yeah,
it's a thing.
No, you still hope that there's no exception.
Yeah.
Okay. More questions? If this is not the case, I thank you again, Yann, in place for everybody, and I'll stop the recording now.